Okay, it's just taking us a few minutes to get online today because I'm handing out exams. So we're getting there. When's our exam, y'all? Next Tuesday. Next Tuesday.
1 to 3?
I believe it is 1 to 3. Is that right? 1 to 3. Okay. Gateway timeout. What? Y'all know that I think I forgot to tell y'all a joke last time. Was it this class? There was a class I forgot to tell a joke to. Last class. Was it this one? Probably. So I owe you 2. Yesterday's was really good, so I'll share yesterday's. What did one snowman say to the other snowman? Do you smell carrots? What wears a red suit and goes, oh, oh, oh? Santa walking backwards. As we wait for Moodle, the entertainment continues. I'm trying to think what else. He was telling a bunch of the car this morning to my mom on the phone. Oh, what do you get when you cross? Wait, I'm going to get this wrong because he kept getting this wrong. What do you get when you cross a Christmas tree and an apple? A pineapple.
Oh, I was thinking apples.
Pineapple. What is it with Moodle today?
Noodles always doing
Well, we just might not have a seating chart today. If that's the way it's going to go, it's okay. Unless somebody, y'all can get on, somebody can get on. I could do it through Google Drive. Let me do it that way.
Well, I can get to it through Drive, yeah.
That's what I can do, right? Go to Drive. More than one way to skin a cat, right? I love cats. I love cats. Don't get me wrong. I used to have cats. I don't know what my password. Oh, really? It's one of those days. Yes, it's me. Exams are here. Okay. All right, so what are we, 4:35? Follow Moodle. What are we, fall? Fall tuition here.
So on this question, I'm pretty sure it should be 4 because it says if at equilibrium B is greater than A.
If at equilibrium B is greater than A,
So
then you're going to shift to the A.
right, but that's saying that's already at equilibrium. That's not to attain the equilibrium.
If you have this reaction, that's this, right? And if at equilibrium you have lots of this, what's going to happen?
Well,
It's going to do this.
right, but it's saying what can be said about the directionality of the reaction if it's already at equilibrium.
No, this is the reaction. And then you just have to look at it and say, are you going to go forward or are you going to go reverse?
Well, see, that's what I was thinking reverse, but since it had said at equilibrium B is much greater than A, that means the reaction is favored in the forward direction. You get what I'm saying? Because
No, it's not.
well, if B is way greater than A, that means that this is converting to this.
Okay, let's they all have it too. So
Oh, okay.
hang on, let me fix this. Oh, I'm not doing the calendar. What am I doing? I'm doing the seating chart. See, this is what happens when I get off track. Okay, here we go. Okay, come on. Rename, what is today? 12/8. instead. /8/21. Okay. All right, so now you can do seating chart. You can click in on iClicker, right? Okay, and then a few people had some questions about exam ones, so we can do them. We can do them real quick. I just need a blank part of the screen. Oh, come on. Okay. So this one. So if here's our reaction, this is the one y'all were talking about. A going to B, right? And at equilibrium, concentration of B is much greater than concentration of A. What's the directionality of the reaction? If you have lots of B, if you have lots and lots of B, what's going to happen to the reaction? You're going to shift it in reverse.
What can you say about the actual reaction, not like what would happen to speed as much as, you know what I'm saying?
That at equilibrium, B is high. I think where the confusion is because it just used the normal
Arrow instead of a double-sided arrow. Well, you don't necessarily know that. Go ahead. If what? Okay, so what is K? Products over reactants. So you're making lots of product. I see where y'all are going. I was looking for something, and I see where y'all are getting confused in this. So I will give you credit. I will give you credit for both. But do you see how it's a shift question? It's not a. I think y'all are just looking at it in a different way, and that's okay.
Like, are you
That's
saying
okay.
where is the shift, or how is it gonna shift? Like,
How is it going to shift?
Yeah.
But that's not okay. I understand. That's what I said. I'm going to give y'all all credit for it. I understand that the way I wrote it is not. Y'all did not get what I was asking, and that's okay. It's okay. That's why we go over these. What's that? Because if you have a case like any given glycolysis, citrate, whatever it is, if you start building up product, what's going to happen? You're going to go in the reverse reaction, so it's no longer going to be a favorable in the forward direction. So this is just an illustration of the feedback that happens in all these different cycles. What do you mean they should be? What should be interchangeable? The arrow only goes one direction.
6.
It's okay. It's okay. It's okay. It's okay. So yes, at equilibrium, you have an equal rate forward, equal rate in reverse, but you're changing the condition, right? You are changing. You're starting out with A going to B. Now I'm saying you're going to go to equilibrium and you're going to have lots of B, right? But it's okay. I will go back and I will give everybody credit for it, okay? Because I can do that. I can go into zip grade and do that, okay? What about? In the following figure, what is the linkage between the two monosaccharide units, and is this a reducing sugar? So what should you have? You should have a 1, and then that is a 1. So 1, 1. So you only had one that was a 1, 1. 1. No, I'm sorry. A 1, 2, because you got a CH2. So that's a 1, 2. So you have a 1, 2, and then you are down and up on the first one. So that is alpha, and you are up and up. No, you are down and down. So you are beta. So you're alpha beta. Okay, so here's your second sugar. Here's your second sugar. Oh no, second sugar is this one. Come on. So here, right, we have a CH2OH. We have a CH2OH. OH, right? And there's that. We'll just draw them both. Okay, did I get it right? Okay. All right, so the first one, right, you find your anomeric carbon. That is carbon number 1 or 2. It's carbon number 1, right? Because there's nothing else attached to the anomeric carbon. So this is a 1, and is it alpha or beta? Because the anomeric carbon is participating in the glycosidic bond. This is up, this is down, so it is opposite, so it's alpha. Okay, now, where's your anomeric carbon in the second? Here's an oxygen, here's an oxygen. There's your anomeric carbon. Your anomeric carbon is either 1 or 2. Is it 1 or is it 2? 2, because this is 1, 2. So this is a 2. Now, what are you comparing for up and down? The 6, the highest numbered carbon outside the ring, and the glycosidic bond. So what do you have? Down and down, which is beta. So beta 1, 2 was A. Yeah? If you did not have a free anomeric carbon, that's the difference between reducing and non-reducing. Non-reducing, you have no free anomeric ERI carbons. Yeah? No, this is anomeric and this is anomeric. They're both participating.
Oh, I thought you meant when you did it has 4 bonds.
No, free anomeric means not in glycosidic.
Gotcha.
What in the world?
It's because of the Zoom.
It wants to grab the Zoom bar.
Oh. I was like, what am I doing? Yes? Good now? Okay. All right. Any else? Anybody else? Yes? Okay, so when we talk about the first high energy compound, the first high energy compound is what? So if you pull up glycolysis, I really should pull up glycolysis. Watch. Where in the world did I put that? So watch. Glycolysis was 9, right? Okay. All right. So where's our first high energy compound that's made? High energy compound made in glycolysis. Is glucose 6-phosphate very high energy? Why do we have to phosphorylate it though? It'll stay in the cell. It'll stay in the cell. It won't get exported out, right? Because what's on the phosphate group? Negative charges. So that traps it inside the cytosol. So what enzyme catalyzes the very first high energy compound in glycolysis? That's the one you're asking about, right? Phosphofructokinase 1, because what? fructose 1,6-bis phosphate is very high energy. That's why we can cleave it into two. ATP is not the only high energy compound that exists. I think y'all see ATP and y'all are just like, okay, what's the first one that makes ATP? Right? That's what you were thinking. That's what you were thinking. But the reason why some of these reactions occur, where is phospho? That's okay. I'm just clicking here, right? So this one, full screen, right, is going to make this really high energy compound that is going to allow us to go on to the next step and separate the two into two lower energy molecules. So ATP is not the only molecule that carries high energy. We can use it in substrate level phosphorylation to make high energy compounds.
Could you explain like what on the molecule makes it itself a high energy compound?
So think about anything. A lot of times enzymes, intermediates, right? Intermediates in enzymes are very high energy. So when you're thinking about stepwise, right, what gives it high energy is its three-dimensional conformation, and that's different for different molecules, right? But this is very unstable. It's a stability thing. High energy usually equates to very, very unstable. And so this, as soon as we make this, we're going to automatically go through the rest of glycolysis. But this is a rate limiting step. We want high energy compound creation to be rate limiting. Because if you do this, you have to put in energy to do it in the form of ATP. So if you don't want to waste energy, you're not going to make high energy compounds. That's why we regulate phosphofructokinase 1 highly. Yeah? Okay. Yes, sir? It is 21. Wait, 21? So we have our enzymes coupled, right? So why do we use shared intermediates when we couple reactions? So when you couple reactions, right, as soon as you make product, you want that product to be made into something else, right? And so if you couple two reactions together, if you just talk about two, right, this reaction is showing 5. But if you just couple two, if you make a product and then use it in the next reaction, what do we call that? A shared intermediate. So the shared intermediates are effective because that product that was made in the first reaction gets used up immediately. You're going. It's not, absolutely not B, right? Because B is dependent on Q, not Q, on K equilibrium. So you're not at equilibrium. That's not at all, right? So if you decrease the value of Q, you are decreasing your number of, but that's not what's happening. That's not what's happening. You're looking at, you have to look at exactly what happens. What are you losing? You're losing the products because the products are being used in the next reaction. You're not just changing concentrations. You have to think about this in not one reaction, but in multiple reactions. You're thinking in just one reaction. Which is what C is. Which is C. What you're describing is C. You limit the product diffusion, right? That allows for the intermediates to channel from one enzyme to the next. Oh, you're on different versions. But it's different versions. Sorry, I always forget that it's different versions. But you're limiting diffusion. Does that make sense? That's exactly what you described to me. Everything that you just said. Okay, any other questions? Good. Okay, and then I will go back and I will add the 2 points. Okay, good. All right, we got to get back to the electron transport chain. Y'all ready for that? Okay, I think that's where I think I wrote on that slide. Didn't I write on that slide? Okay, so we talked about the Q cycle, right? Okay, so once we're done with the Q cycle, what has picked up our electrons? Cytochrome C. Cytochrome C is carrying how many electrons? One at a time. Please make sure those masks are up, everybody. One at a time, and they are carrying them to what next component in this cycle? Complex 4. Complex 4. Okay, so if we look at cytochrome structure, so all of the cytochromes, right, are really small molecules. Some of them are embedded in membranes and some of them are not. So the type A and the type B, these are embedded in membranes. Go look, take a real quick look at complex 1, complex 2, complex 3. Do you see anything that has a B and then a subscript that's involved in the chain of passage of electrons from one place to another? Do you see it? So if you go back, see this, the B, right? There are different versions of them, but these are hemes, okay? So the B, for example, you can find in complex 3. Example is in complex. It's not the only complex it's in. What other complex did you find it in? Did you look? Anybody? It's in other complexes. Type A is also in one of the complexes, but this one, if we go forward, right, and we look at complex 4, we can see some of the A-type hemes inside of complex 4. So this is in complex 4. So these are membrane bound. They're stuck inside of the membrane. But type C is unique. Why is type C unique? When we look at this picture, is this cytochrome C embedded in the membrane? No, it is not. It is free to float around. So this one is a stable monomeric unbound protein. And these have been around for a really, really long time. You look at conservation of amino acid sequences, and these are super, super insanely highly conserved, right? And so whether or not this is oxidized or reduced, what are you looking at? This iron containing what group? It's a heme, right? So they all have an iron containing heme. So when this is oxidized, what is it? We just show it as Fe. So what is it if it's oxidized? If it's reduced, Fe, no, oxidized is 3+, reduced is 3+. Yeah. Okay. So this is the type that is going to actually pick up the electrons and transport it from one complex to another, because this is the only one that's free. So cytochrome C is going to take over those electrons, but it's going to have to do it in 2 trips, right? So that's why you need 2 cytochrome C molecules in order to get to complex 4. What is the name of complex 4? We're only naming one transport molecule, right? Cytochrome C. And this is the oxidase because this is the end of our chain, right? We're going to drop off those electrons. They're going to go through a little bit of a path, and eventually those 2 electrons are going to be given to what? Oxygen, right? So for every 2 electrons we're going to have to pick up 2 hydrogen atoms, but can you have half a molecule of O2? No, not really. We show this as a half reaction, right? We show O2 as half, but what do you actually need in order to reduce 1 molecule of O2? You need 4 electrons, you need 4 protons, and you're going to make 2 waters.
Is the picture showing it happening just twice, or is that just showing how one time when cytochrome do it?
This is saying that for every 2 cytochrome C, right, you get 2 electrons. Those 2 electrons go all the way down here. Those 2 electrons are required to reduce half of an oxygen molecule. So for every 2 electrons, you're going to have 4 hydrogen atoms that are going to be attached to your oxygen molecule. So you'll make 1 molecule of O2 for every 2 electrons.
So even though it happens, it carries it on at a time, when it comes back a second time, that's when the process starts?
Correct. So at the same time, right, this is a pump. We're actually going to move 2 more protons from that intermitochondrial matrix to the intermembrane space, right? Okay, so if I ask you, what do you need at the very end in order to reduce 1 oxygen molecule, are you going to say 2 electrons? No, you're going to say you need 4 electrons, you need 4 protons, plus what? 1 molecule of O2, which will give us 2 molecules of H2O. 2, if I could do this. But why do we start at why do we even say 2 electrons? It's not really 1 turn, 1 what? It's 1 something. Yes, go back another step. 1 molecule of what enters the electron transport chain? 1 molecule of NADH drops off how many electrons? 2. So basically, when we talk about all these figures in the electron transport chain, we're talking about if it's 1 molecule of NADH, or depending on if it's a different complex, FADH2, right? So that's what you have to kind of think about. That's why these cartoons are drawn this way, because we're really tracking what happens with 1 molecule of NADH, with the electrons from 1 molecule of NADH. Okay. All right. So you've pumped hydrogen ions, right? That was the whole name of the game, was to build up hydrogen ions in the intermembrane space. Did we accomplish that? Yes. For every molecule of NADH, how many protons did we pump to that intermembrane space? 10. We pumped 10. So now that creates 2 things, right? That creates a change in pH, and it creates a change in membrane potential, right? Which one is more important? Guess what? We're going to calculate it today. We're like, oh man. All right. So when we talk about this, right? Really, we're talking about trying to calculate delta G, right? That's the whole name of the game. And so before we talk about delta G as it is normally, let's talk about standard delta G. Delta G not prime. Do we have a formula for delta G not prime that will relate it to our standard standard potential for transferring electrons? Remember this standard reduction potential? Do we have an equation for that? Yes, negative N, Faraday's constant, delta E naught prime, right? Okay, so I could use this to calculate delta G, This would be optimal, right? If I calculate delta G naught prime, this is the most amount of free energy I'd be able to get. So I have to go in and first calculate my standard reduction potential. What's my formula for standard reduction potential? It's the acceptor. We didn't do the math for this, but it's just the acceptor minus the donor, right? So if I take the whole electron transport chain, I want my acceptor to be the end. I want my donor to be the beginning, right? So what's the end? This really should be primes. Oxygen, it's 1/2 O2. Okay, now what? Where's my beginning? What did I start with? NADH. Good. So where could you go to get these values? Remember that table in the very beginning that had all the reduction potential values for each of them? So if you go look them up, right? Oxygen is the most positive at 0.82, positive 0.82, minus, and it is negative 32 volts. Okay, so what's our standard reduction potential between these two molecules? What do we get? 1.14 volts, right? All right, now we can take this 1.14 volts and plug it in right here, right? So I can say delta G naught prime is equal to, okay, what is the end? It's the number of electrons. So how many electrons did we move? 2. F is Faraday's constant, 96.48 kilojoules per volt mole. Okay, and then delta E naught prime is, what did we say? 1.14. Okay, volts. So what do we get? Check my math. What do we get? Should be negative because it happens, right? It's got to be negative. So we'll round to 220, right? 220 kilojoules per mole. Okay, that's standard, right? But do we really want standard? No, not really. So we want to compare this value that we got for standard biochemical conditions for actual, right? This is actual. This is standard. Standard is our basically optimal conditions on everything. This is the max you will ever get mathematically. But in real life, are we really going to get this much energy? No. So let's plug it in to this one now, right? Can I just move the rest down? I really love to just move all that stuff down. I'm so used to being able to do that. But I'm going to go down, but I'm going to plug into this formula. Okay, y'all ready? Okay. Some of these values I have to give you, you don't know offhand. So delta G is equal to R. What R do we want to use? 8.314 times 10 to the minus 3. So what are my units? Kilojoules per mole K, right? Okay. Now what's my temperature? 37 degrees Celsius. So that translates to, this is body temp, 310 Kelvin. Like this is one thing, if you're doing any chemistry within the body, you should just automatically think 310. You should just, like, that's should just be there. Okay, now Ln, the concentration of the hydrogen ions on one side versus the other side. I'm going to tell you that that ratio is about 0.75. okay? That's a number I'm giving you. After the electron, as the electron chain has been working normally, that should be your difference, okay? So this part of the equation is really based on the concentration difference between the two sides, right? So that concentration difference results in a change in pH, right? Now, the other half of the equation is going to be dependent upon the membrane potential. So if we look at the other half, it's it's Z. What is Z? Charge is 1. It's 1. Okay. What are we, Faraday's 996.48 kilojoules per volt mole. I always want to write mole, volt mole. Okay. Now, our membrane potential is what we have to really worry about here. This value I would have to give you, right? You're not going to know that offhand. And based on this concentration difference, you can calculate membrane potential, but you're not going to have to do that calculation. I would always give that to you. So this would be negative 0.150. So this part of the equation is due really to what? Change in membrane potential. So calculate and tell me, what are these two sides of the equation equal to? Because what I want to know is I want to know what's actually driving ATP production. Is it my change in pH or is it my difference in membrane potential? And this mathematically will tell me that. So what do you get for the first half? What do you get? 0.74. Wow, that's not a whole lot, right? And what do you get for the second part? It's also negative. 14.47. And both of these are in kilojoules per mole, right? So which one contributes more to the delta G value? Our membrane potential, right? Okay, so what do we get for our standard delta G? Negative 15 to 1. Okay, so that's really what I want you to think about when you're thinking about how are we going to use this proton pump to drive ATP synthesis. What's the difference? The difference is really going to be that membrane potential. That is what is going to drive and give us that really large negative delta G value that's going to allow this reaction to go and for the electron transport chain to be spontaneous, right? It's not really a pH difference. It's really a membrane potential difference, but both are caused by those hydrogen ions being pumped. I think that's an important distinction. Yep.
Is the C2 the hydrogens in the intermembrane?
Yes. Yes. It's matrix to intermembrane space. Yes. Okay. So we've pumped hydrogen ions, right? The next thing is that we actually need to make ATP. Thank God we're going to finish this semester. At least we're going to make ATP, right? So when we look at the ATP synthase complex, right, it was originally designated as complex 5, but we don't really call it complex 5 anymore. We call it the ATP synthase complex because we know exactly what it does. So we're going to kind of talk about it in two ways. We're going to talk about it structurally, and then we're going to talk about it functionally. So if you just look at the structure, you have two major components, whether you're talking about something in a complex system, and by complex, I'm saying yeast, right? Compared to bacterial, we're a whole lot more like yeast than we are bacterial, but it's basically the same thing. So what you'll see is that there is part of your ATP synthase complex that's embedded in the membrane, and then you have part of it that is actually inside the mitochondrial matrix. And it's different inside of bacteria because bacteria, it's actually embedded in the cell membrane. But this is what we're really going to focus on. So we have our F1 complex that is down here at the bottom. And when you look at that F1 complex, this is actually going to be the part of the molecule that has the catalytic activity or the enzymatic activity that will convert ADP and PI into ATP. So this is our catalytic activity down here. The F0 component is inside, embedded in the membrane, and it is actually functioning as a channel, right? Because we're going to harvest the energy that's in that difference in protons, right? So the protons have to come through the ATP synthase complex. That is in the F0. And then once they come through, it's going to induce a conformational change in the intermitochondrial matrix portion of the molecule, and it's going to basically turn the rotor, and you're going to generate ATP. So the catalytic is basically in the matrix, and then the channel is in the intermembrane space. Okay, so if you take a look, you can actually break this down into 3 major components. So in that F1 complex, you have what's called the headpiece. The headpiece is what contains the catalytic subunits, right? And each of these individual A's and B's, alphas and betas, right, are going to bind different things, right? They're going to have to bind ADP and PI. Then they're going to have to induce a conformational change. They're going to phosphorylate the ADP and then release ATP. So that's the headpiece. Now, if you look at the part of the molecule that allows the hydrogen ions to come through, that's going to be here. So do you see how hydrogen ions come in and they go out? This is called the stator. So this basically holds everything together. So see how the arm kind of protrudes into the matrix, and then it holds the headpiece in place. So this gray piece is basically, you see how it's not one straight line? It's actually 2 half channels. So hydrogen ions come in, and then where do they go? They're actually going to go into this blue portion of the rotor and cause the rotor to change. And then once the rotor changes, those hydrogen ions can come out on the other side of the membrane. So when this blue thing turns, this is called the rotor. It actually has a piece that extends right smack down in the middle of the headpiece. So it actually bumps along on the different parts of the headpiece, and this is what induces the conformational change. Every time that rotor hits one of those subunits in the headpiece, it changes the conformation of the headpiece. And that's what actually drives ATP synthesis. You really need to watch the animation. It's really awesome. You should watch the animation. Okay, so how do we get that conformational change in our ATP synthase, right? So first, the protons have to go through that channel. The channel is in the F0 component, right? Then that, as those protons flow, why is that proteins? It's supposed to be protons. Y'all change that to protons. Protons, right? Hello. As the protons flow through the F0 component, right? Then they're going to interact with the rotor, and the rotor is going to induce those conformational changes. We call them 3 different conformational changes. We call it loose, tight, and open. Okay? That rotor is going to rotate 120 degrees. And in that 120 degrees, ADP and PI will bind and we will form ATP. Certain things happen in each conformation of that headpiece. So only when you're in the open position is ATP going to be released. So as soon as we make ATP, you're going to release the hydrogen ions from the other end of the channel. And then we're going to lose that proton gradient, and the protons can go back and re-go through the electron transport chain, right? Okay, so let's look at what actually happens. So we'll kind of go through it in 3 different stages. So if you look here, we're going to pick one of these subunits to look at, only one of them. We're going to look at the B1 subunit. So if we look at the B1 subunit, It's in the open conformation. So what do we say happens in the open conformation? ATP is released. So by the time we make this circuit again, we should be able to release another molecule of ATP. So when the ATP is released, now we have an open pocket. That open pocket allows ADP and PI to come in and bind. So now in the loose conformation, we now have ADP and PI bound. We're going to go through another turn, right? So do you see if you look in the middle, you see this? This is this orange piece with the circular arrow in it. So if you look at that, that is this rotor right here. This is the gamma subunit of the rotor. So do you see how the rotor, it's long on one side, but not on the other, right? So it's going to bump along. I can look at it. It's going to bump along and hit each of the different subunits and induce that conformational change in the subunit, right? So now we bound ATP and PI in the loose conformation. Now we're going to, that rotor is going to turn again. And when that rotor turns again, what's going to happen to this B1 subunit is that you're actually going to have that phosphorylation event. And you're going to have the inorganic phosphate attached to the ADP. Now what substance do we have bound to beta 1? ATP. We're going to undergo one more conformational change, and it's actually going to open up. And what are we going to release? ATP, excuse me, right? But now, we only tracked one subunit at a time. But how many beta subunits are there? 3. And there are 3 different conformations. So at any given point in time, each of the subunits is in a different conformation. So at each place, do you see how here in this model, we're losing an ATP from beta 1? Go to the next one, we're losing an ATP from beta 3. Go to the third position, and we're losing an ATP from beta 2. So every time that rotor moves, you generate an ATP molecule. It's just coming from a different beta subunit, right? And that's because the rotor is hitting the different subunits, and that causes the conformational change. So every time the rotor moves, it requires 3 hydrogen ions to be translocated through that pump. So if I go through this one time, how many hydrogen ions was that? How many? 3, 6, 9. How many do I get from one molecule of NADH? 10, right? So it isn't perfect. It isn't perfect, but it takes on average 10 to do this. So it takes a little bit more. Do you see how they have the little squiggly 3, the little squiggly 3, the little squiggly 3? Yeah, that's because on average it takes 10. So there was this really amazing experiment that was done with ATP synthase, showing that that motor actually moves, right? Because the question was, okay, we know it's undergoing conformational changes, but we don't really understand how ATP is actually being made. How are we inducing conformational changes that make ATP? And so what they were able to do is they basically attached the catalytic subunit of ATP synthase to a solid surface. So like a slide, a glass slide, right? And then what we do is we attach an actin filament, right? It's very strong, very straight cytoskeletal protein, right? And we attach it to the rotor exactly where this blue part of the rotor is supposed to be, right? And so what we can see is as we turn the rotor, so depending on what direction you turn the rotor in, instead of creating ATP, if you turn it in the opposite direction, you are actually going to hydrolyze ATP. So you can put in solution on this glass slide, ATP. You run this motor by turning the actin filament. You basically put a magnet on the end of the actin filament, and you take the actin filament and you spin it around in the opposite direction that it normally spins in, which is a cool experiment in and of itself. And then you will see that because it goes in the opposite direction, it induces the opposite conformational changes, and you will actually hydrolyze ATP. Isn't that awesome? That's so cool. So this was really great proof, right, of actually what this ATP synthase molecule can do.
Does the hydrolysis have like a purpose? Like, does it use ATP for other purposes?
No, this was just like a proof of purpose. Like what is this actually doing? So if I isolate just the part of the rotor that induces the conformational change on the beta subunits of the catalytic head, is that all I need to hydrolyze or go in the reverse to make ATP?
So it doesn't have an actual purpose in the actual cell.
In the cell, no, this never occurs in the cell. This was like, okay, we think this is what's going on in the cell. So let me isolate just the pieces of the ATP synthase complex that we think are required to actually make ATP. So let's get rid of the hydrogen ion pumps, and let's try to drive this motor in a different way. And the different way that they drove this motor was to attach the actin filaments with a magnet on the end and then crank it like a motor, like a molecular motor. So cool. And you can actually do that. And then you do the opposite process. Instead of making ATP, because we crank it in the opposite direction, we can hydrolyze ATP. And we can monitor levels of inorganic phosphate, right? How many times did we do that in lab this year? Yeah, a bunch, right? A bunch. I actually had to think about it. We can monitor the amount of inorganic phosphate in any solution. That's an easy experiment. We do that here. So you can actually monitor production of inorganic phosphate. It's really cool. All right. So I'm not going to start this, but I do want to talk. Don't pack up yet. Don't pack up yet. I want to talk about the very last section, right? There's the try it yourself questions at the end, right? I want to do that with y'all. So the question is in there, what's the role of coenzyme Q in the ETC? Right? So we talked a whole lot about Q, did we not? Yesterday a little bit, yesterday a lot, today a little bit. So don't look at your notes. Try, you can write it down, but try to think about, why is Q important? Throw it out there. Anything. It's not the hydrogens.
Or electrons.
Yes, so Q does transport. Q transports electrons. We'll say electron pairs, right? To what? Complex what? 3. Okay, that's very true statement. Why is Q so important in the electron transport chain? You're just linking something to the next, right? How many things bring electrons to Q? A lot. Let's see if we can list them all. What brings electrons to Q? In order, like, let's say I'm NADH. That's the beginning of the cycle. What if I'm NADH? I go from complex 1 and I give up my electrons to Q. That's true. What if I am FADH2? I could go directly from complex 2 to Q. There are 2 other FADH2 transporting molecules. What are they? Transporting systems. There's a shuttle, glyceraldehyde 3-phosphate shuttle. Right? Where do we get those FADH2s from? Glycolysis. There's another one. Fatty acids, right? Anybody know what that one's called? GTFQ, right? So what is the role of Q in the electron transport chain? It's ETFQ. Sorry, ETFQ. So what's the role of Q? It's an electron transport system, right? It's embedded in the membrane, so it's hydrophobic. It's not going anywhere. Like cytochrome C can kind of be off going anywhere, but Q is kind of stuck in the membrane. But Q is this amazing protein that links all the different ways electrons can enter the electron transport system to complex 3, right? Should you try those end of section questions? Yeah. Okay. So if Q picks up electrons, let's say it doesn't matter where Q gets those electrons, what happens to Q? Converted to what? QH2. QH2 goes to complex 3. But what's so important about bringing it to complex 3? What can complex 3 do that's super important? Complex? Not yet. Not yet. So here is where we separate, right? To separate electron pairs, right? So Q is carrying 2 electrons. Then we convert that to cytochrome C, which carries 1 electron at a time. Right? That's why Q has to bring everything to the same place. That's why Q brings everything to 3, so that complex 3 can separate those electron pairs into single electrons. Then those single electrons can go where? 4, where we end and we reduce oxygen to water. And in the process, which we didn't list here at all, what else happens in concert with moving these electrons? Translocating protons from the matrix to the intermembrane space. So what's more important about those electrons in the intermembrane space? Their change in pH or their membrane potential? The membrane potential. Mathematically, you know that now. Isn't that kind of nice? Would you like to know the math behind it? Not today. That's okay. That's okay. All right, any questions? I will see y'all on Tuesday. No, as far as we got is as far as we got. Don't forget exams.
There you go.
Thank you. Thank you, thank you, thank you, thank you. Make this efficient exit. All right.
So you said about like question number 6. Was that already added or is that going to be added?
That's gonna be, well, it's going to, I'm gonna go in and if you got it right, you got it right. But I'll go in and
Give everybody
yeah.
who got it wrong.
Yep.
Okay. That makes sense.
Uh-huh. Yeah, she's gonna ask me one first, but then
Okay.
I'm trying to make sure everybody who wants to get out can get out.
Okay. I'm sorry.
That's okay. All right, oh wait, I got something on the chat. Hold on. I'm sorry, I missed announcements. What did I say in announcements, y'all? At the very beginning of class? I don't remember, but I'm gonna post the recording and you'll be able to go back and watch the recording. I think it was a reminder, just whenever exam on Tuesday, yeah, from 1 to 3.
In here?
In here, it'll be in here. And I think that was all I had said in my jokes, but I think that was it.
Is there gonna be like a seating chart?
No, I'm asking everybody to try to sit in the similar seats as they have been all semester, so that we don't have to have a seating chart that day. That makes sense. All right, cool. Any other questions? Oh no, everybody left. Okay, well, that's a good way to